Hi, this is Dr. Brandon Gehrig from Northern Michigan University, and today I will be discussing a Great Lakes Fishery Commission funded project related to the trophic overlap of lean and Siscowet lake char across their trophic ontogeny. Increases in juvenile Siscowet abundance have been observed in summer surveys that index lean lake char recruitment in Lake Superior. And this has raised the concern among managers that juvenile Siscowet may compete with leans, potentially influencing the maintenance of rehabilitated lean lake char stocks, along with hurting valuable recreational fisheries. Now, if competition is occurring as a result of co-occurrence between juvenile lean and Siscowet lake trout, it's important to determine the extent to which they co-occur, and also whether they're consuming similar prey items, and whether any overlap has the potential to impact fitness directly with respect to reduced growth or survival. So with this in mind, the objectives of the study were to characterize spatial, dietary, and isotopic overlap of lean and Siscowet lake char across their trophic ontogeny. Now, past diet studies support the assertion of both niche partitioning and overlap among lean and Siscowet lake char, but they largely focused on large adult fish. So for instance, Harvey and Kitchell in 2000 found little evidence of trophic overlap between adult ecotypes, and they proposed that this was a result of both habitat and resource partitioning. However, a newer study in 2017 by Heidi Swanson's master's student found that there was extensive isotopic overlap between lean and Siscowet lake char, but the effects of size or age were not evaluated. So with that in mind, our study is really seeking to kind of fill this data gap to try to understand how lean lake char are partitioning resources or overlapping across their ontogeny. To do this, we sampled across 6 ecoregions of Lake Superior along the South Shore, ranging from Minnesota waters to Wisconsin to several ecoregions within Michigan. Collections were made for our study based upon gill netting and trawling efforts during the summers of 2019 and 2020 as a part of standardized monitoring programs employed by state and federal agencies. These surveys focused primarily on waters that were less than 80 meters in depth. And any fish that were captured, particularly lean and Siscowet lake char, we collected their stomachs for gross examination of gut contents, along with taking a small isotope sample so that we could quantify carbon, nitrogen, and sulfur isotopes. So to look at our first objective of how juvenile lean and Siscowet lake trout are distributed, I'll orient you to the graph. We have several length bins on the bottom that are divided by millimeters, and then we have the geometric catch per unit effort on the y-axis. And you'll note that this is a tri-paneled plot that is split amongst depths ranging from 20 to 39 meters, 40 to 59 meters, and 60 to 79 meters in depth. So our first finding was that the catch per unit effort was highest for lean lake char in waters that were less than 60 meters in depth. And we found that catch per unit effort declined quite substantially at depths that exceeded 60 meters in depth. We also found for Siscowet that catch per unit effort was generally lowest in waters that were less than about 40 meters, and that Siscowets tended to dominate catches in waters that were greater than 60 meters in total depth. So based upon this analysis of survey data, we find that cohabitation is highest, particularly at depths between 40 to 60 meters. But it is important to keep in mind that most of Lake Superior's deep water habitat. In this plot, this is a plot of total length by depth of capture, and it contains data from a broader survey effort than just our current study. And as you can see from this plot, leans of all sizes rarely inhabit depths that are greater than 60 meters in depth, and juvenile Siscowets located in this area below this kind of red box rarely inhabit waters greater than 240 meters. And most adult lake char, so these are going to be fish that are greater than about 500 millimeters, spatially partitioned by depth. And this is proposed to be a self-regulating mechanism against direct competition for resources. So why would juvenile fish overlap in waters that are less than 80 meters? Well, high spatial overlap at small sizes may actually be the result of where spawning habitat is located for both lean and siscowet, rather than any sort of other biological mechanism. So prior data has suggested that siscowet spawn at intermediate depths, and by putting their eggs and having their juveniles reside in intermediate to shallower waters, would allow juvenile siscowet to begin their lives in more productive habitats and potentially minimize the risk of cannibalism, albeit overlapping with juvenile leans. For our second objective, we found that diet composition varied by ecotype and length bin. Now you'll see a series of diet composition plots where length group in millimeters is on the x-axis and proportional biomass is on the y-axis for both ecotypes. Note that food web items are oriented from the top to bottom based on their relative position in the food web. So we have invertebrates like mysis, diaperia, and mollusks towards the bottom, next followed by sculpin and burbot are benthic fishes. We have stickleback, smelt, corygonids, various salmonids and unidentified fishes making up pelagic fishes in the open water column. And then we have a surface category that's confined that corresponds to terrestrial insects. Now, overall take-homes from this data are that both ecotypes are opportunistic predators and that omnivory is common across their trophic ontogeny. This is really driven by widespread consumption of mysis, terrestrial insects, and fish at all size classes. Now, dietary diversity does increase with increased lake char size, and this is likely reflective of both the habitat shifts that both ecotypes appear to undergo, and also a result of increased gape size allowing consumption of larger prey items. When we look specifically at small char, we found that lean diets had nearly equal contributions of fish, like rainbow smelt and sculpins, along with invertebrates such as mysis, terrestrial insects, and diaperia. Now, Siscowet, in contrast, consumed a solely invertebrate diet based upon our data, with mysis being the key prey item for Siscowet less than 300 millimeters. Now, it is true that lean appear to have a greater dietary diversity than Siscowet, and this appears to be driven by the increased contributions of both benthic and pelagic fishes relative to the primarily invertebrate-based diet that we observe Siscowet to have. Now, if we shift to medium char, we find that piscivory becomes much more common for larger fish that are exceeding 400 millimeters. Lean shifted to piscivory of primarily pelagic prey, where rainbow smelt and corygonids make up the most commonly consumed fish items. But it's important to note that terrestrial insects and mysis remain approximately 25% of the diet by biomass. Now, when we consider Siscowet diets, we find that their consumption of mysis is reduced, and it is greatly replaced by contributions of benthic sculpins along with pelagic smelt. But we also find, like leans, that terrestrial insects and mysis make up a greater than 25% of the diet. So this is suggesting that they're capable of integrating prey items from the entire water column, even at these smaller sizes. For large char, we find that leans continue to feed extensively upon rainbow smelt, but several other prey items are represented. Importantly, we still find significant contributions of both terrestrial insects and important invertebrates, including mysis. When we consider Siscowet greater than 500 millimeters, we find that they exhibit high levels of dietary diversity. This is relatively speaking because we're in Lake Superior. But we find significant contributions of both terrestrial, pelagic, and benthic prey resources, with terrestrial insects and mysis remaining important invertebrates, along with fishes like sculpin, burbot, smelt, and small proportions of deep water corygonids. We also adopted a graphical method developed by Amundsen et al. To characterize prey importance and feeding strategy of lean and Siscowet across their ontogeny, and to compare prey-specific abundance to frequency of occurrence. And we define this graphical method based upon two key vectors. The first vector defines prey importance, and this is diagonal that originates in the lower left quadrant and moves to the upper right quadrant, which provides a measure of prey importance. Here, rare prey items occupy the lower left quadrant, and dominant prey occupy the upper right quadrant. The second vector, defining the between or within phenotype contributions to niche, runs from the upper left quadrant to the lower right quadrant. And populations in which individuals specialize on different resource types are going to occur in the upper left quadrant. Okay, so if populations are being specialized, they'll be in that upper left quadrant. Based upon our graphical analysis, all lake char, regardless of ecotype and size class, had high between phenotype variability in their diet, as indicated by prey items being concentrated in the upper left quadrant of the figure, as denoted by the red boxes. And based upon interpretation, this would suggest that lake trout are specializing on particular prey items. However, we interpret this rather as the result of opportunistic feeding behavior in a resource-poor environment where prey are heterogeneously distributed. For our third objective, we also examined stable isotope relationships for lean and siscowet across their ontogeny. Stable isotopes provide us with a time-integrated measure of resource use and have been useful in inferring niche overlap in many fish species. We'll now walk through a series of kernel density ridge plots to show us how the distribution of isotope ratios varies across size classes and isotopes. The black vertical line that is present in the graph represents the median isotope ratio for a given length ecotype combination. We'll first begin with carbon, and carbon provides us with information on the primary energy source which supports the food web. Based upon our analysis, lean and siscowet carbon signatures move only slightly, but they shift in opposite directions of about 1 per mil in the opposite direction as they increase in size. Based upon a prior study, it was observed that the carbon signature of rainbow smelt was depleted relative to other pelagic fish. So it's possible that the slight shift in lean is reflective of increased consumption of smelt, which is something that we see in our diet study. When we examine nitrogen, nitrogen provides us an indication of trophic position, but also can provide an indication of the degree to which foraging is occurring at deep depths. For leans, we observe a 1 per mil shift, which we attributed to larger fish feeding upon larger prey items, which have higher N15 signatures. However, it gets interesting for siscowet. We observe a strong 3 per mil shift in nitrogen at around 300 millimeters. This increase in nitrogen is related to predictable increases in nitrogen with depth. So we think that this increase in nitrogen in siscowet is reflective of an ontogenetic shift to feeding at greater depths on prey items like benthic sculpin and burbot, which we saw present within our diet analysis. Note that the median N15 value for siscowet also declines at sizes greater than 500 millimeters. And we hypothesized that this decline in N15 actually is reflective of increased vertical movements of siscowet in the water column and continued incorporation of lower trophic level prey items, including both mysis and also terrestrial insects. When we also examine change in sulfur isotopes with size, and we find that the results closely match that of nitrogen, where in siscowet, we particularly see a directional shift at around 300 millimeters, which we are attributing to increased benthic feeding at deeper depths. Now, isotope data can also be used to estimate isotopic niche overlap. And in this particular plot, on the y-axis, we have nitrogen isotope, which indicates our trophic level. And the higher the nitrogen value, the higher the trophic level. And on the x-axis is carbon or sulfur. And this relates to basal energy source of production. Each plot in the graph represents the isotopic signature of an individual lake char, and the ellipses represent the 95% credible regions of a given ecotype size combination. And the table represents the probability and credible intervals that a randomly chosen siscowet will be located within the niche of lean lake char. So with that background in mind, we observed high levels of isotopic overlap of lake char less than 300 millimeters. But as lake char increase in size, isotopic overlap is diminished. And we can see this very clearly where we have high probabilities of overlap for our size classes of less than 299 millimeters and and between 300 to 399 millimeters. And then we see gradual declines in that likelihood of Siscowet occupying the niche of lean lake trout at those larger size classes. So we can very clearly see this onogenetic shift that is happening and it's manifest within our isotope data. Do lean and Siscowet lake char occupy similar habitats? Lean and Siscowet appear to be sympatric at smaller sizes, and this may optimize access to key prey resources like mysis as juveniles and reflect the relative distribution of both spawning habitat and also habitat that's suitable for juveniles. So why do juvenile Siscowet occupy shallower waters relative to adults? Well, shallower habitats likely facilitate growth and allow for onogenetic increases in lipid storage, which then allow juvenile Siscowet to make a go out of it in the depths. So do lean and Siscowet lake char eat the same thing? While it's true at small sizes that diets and niches of both of ecotypes are more similar, this may simply be a function of the fact that we're dealing with a relatively simple food web in Lake Superior. We do find some evidence of benthic pelagic partitioning of smelt and sculpins between lean and Siscowet, particularly for fish less than 400 millimeters. We also find that Siscowet appear to transition to piscivory later than leans. We also find that depth and diet vary strongly with increased size. So while they do eat the same thing at small sizes, there's increasing partitioning at large sizes, really beginning around 400 millimeters. And last, is resource overlap in lake char ecotypes negative? Well, this overlap appears to have limited effects on growth based upon some other data that I'm not presenting here today. And really, negative effects of overlap can be mitigated by both the opportunistic feeding strategies coupled with the abundance of key invertebrate food resources. And the partitioning that we see at larger size classes appears to be very pronounced due to significant onogenetic shifts. And with that, I would love to entertain any questions. Thanks so much.